Beta thalassemia, an anemia prevalent primarily in some Mediterranean countries. The disease is due to faulty protein, which forms part of hemoglobin in red blood cells. The protein is called beta globin. If no or barely functioning beta globin is made, the lifespan of the red blood cell is shortened, resulting in anemia. In different patients, small defects in genetic material have been found, resulting in errors in splicing process and thus in the synthesis of poorly functioning beta globin. In the upper part here, normal splicing beta globin in the globin gene is damaged. If the globin gene is damaged markedly by the arrow, it may, for example, lead to the formation of larger than normal oxone during splicing of P, or to formation of completely new oxone like NC. Here the beta thalassemia, defective splicing causing beta thalassemia, a normal beta globin gene is presented in A and 2 mutated gene that result in beta thalassemia. The arrows may mark the position of point mutation. The interrupted line denote the segments that are being joined during the splicing process. In the healthy individual, 3 segments are spliced, as shown in A here. A in one of the thalassemia case, in an unusual long third segment is formed, the B, while in second one an extra segment is produced as in C. Another example showing the connection between disease and the organism secondary to oxone and intron: chronic myeloid leukemia, a type of cancer of the blood. Characteristic for this disease is the presence of in tumor cells of a special shortened chromosome called the Philadelphia chromosome, named after the city in which was discovered. This chromosome has arisen in the white blood cells by fusion of one end of the chromosome 22 to one end of the chromosome 9. At the breakpoint, a large portion of the cancer gene has been joined to another gene. Here are thus dealing with 2 genes which are now coded into one single RNA molecule. During the splicing process, exons from the 2 genes are spliced to form an RNA molecule that specifies the synthesis of new protein, so-called fusion protein. This new protein gives rise to leukemia. Messenger RNA splicing of exon and removal of intron. This is a process discussed before. And this is a full detailed description of the process. Splicing occurs, transcription by RNA polymerase 2, adding 5 cap, 20 to 30 nucleotide of pre-messenger RNA made. Here the cap, oxone, intron, exon, intron, and the poly A, tail, trailer, RNA splicing, intron removed, and the protein coding sequence, translation, initiation of messenger RNA transcript, active spliceosome splicing, the mature messenger RNA is formed. Exists intron sequence in lariat shape, still complexes with a small nuclear RNA. Post-transcription modification of messenger RNA editing adds or delete nucleotides from pre-RNA or chemically altered base, so messenger RNA bases don't match the DNA sequence, can result in substitution, addition, or deletion of amino acid relative to the DNA template, generally cell or tissue specific. Example: protozoa, slime molds, plant, organelles, mammals. Genes that don't code protein also are transcribed. Ribosomal RNA catalyzed proteins. This is by facilitating the binding of transfer RNA and their amino acid to messenger RNA. Transfer RNA transport amino acid to messenger RNA for translation. Small nuclear RNA combine with protein to form a complex used in RNA processing. The synthesis of ribosomal RNA and ribosome cell contains thousands of ribosomes, consists of two subunits, large and small, in prokaryote and eukaryote, in combination with ribosomal protein. E. coli 7S model, 50S subunit, 30S subunit, mammalian 80S model, 40S subunit. DNA regions that code for ribosomal RNA are called ribosomal DNA. Eukaryotes have many copies of ribosomal RNA gene randomly repeated. Synthesis of ribosomal RNA and ribosome transcription occur by the same mechanism as protein coding gene, but generally using RNA polymerase I. Ribosomal RNA synthesis requires its own array transcription factor. Coding sequence for RNA subunit within the ribosomal DNA genes contain internal ITS, external ETS, and non-transcribed transcribed spacers. ITS units separate the RNA subunit through the pre-RNA stage, where ETS are leaved out and the ribosomal RNA are assembled. Subunits of mature ribosome are bonded together by H bonds, finally transported to the cytoplasm to initiate protein synthesis. Here mammalian example of 8S ribosomal RNA, 60S subunit, 40S subunit. Ribosomal DNA repeat unit. Synthesis of transfer RNA. Transfer RNA gene also occur in repeated copies throughout the genome and may contain intron. Each transfer RNA about 70 to 80 NT in length has a different sequence that binds a different amino acid. Many transfer RNA undergo extensive post-transcription modification, especially those in the mitochondria and chloroplast. Transfer RNA form clever leaf structure with complementary base pairing between regions to form 4 stem and loop. Loop 2 contains the anticodon which recognize the messenger RNA coding during translation. Same general mechanism using RNA polymerase III promoter, unique EFS plus post a transcriptional modification from the pre-transfer RNA. The models, anticodon loop, and the crystallography, and this is the diagram of the structure of the messenger RNA. Synthesis of small nuclear RNA form complexes with protein used in eukaryotic RNA processing. Splicing of messenger RNA after intron are removed, transcribed using RNA polymerase II and III.